The seventh recent discovery that has changed the debate about design is that the mechanism of random mutation and natural selection has been shown to have severe limitations. It does not have the tremendous creative power once ascribed to it in the past. Discoveries 3 through 6 revealed aspects of living things that were previously unimaginable, that the sequences of enzymes are vanishingly rare, that the simplest living organism has about 450 genes, that those 450 genes do not just code for random proteins, but rather they produce a digital information processing system, and they code for molecular machines and sophisticated software algorithms, and that all of these have aspects characterized by irreducible complexity and hierarchical coherence, as in engineered systems. In other words, these are immense informational discontinuities. But at the same time that these discoveries were being made, showing life was far more complex than ever imagined, new insights were accumulating regarding the limitations of the mechanism that was supposed to explain the complexities of life: random mutation and natural selection. This is the blind watchmaker, the mechanism that is supposed to produce things that have the appearance of being designed for a purpose. In the minds of many scientists, these two trends, one showing that life is more complex than ever imagined, and the other that random mutation and natural selection is severely limited, has led to a crisis in the field of evolutionary biology. The discovery itself is not controversial, but conclusions among scientists differ. I'll speak about that a little while later, but first I will explain some of the new insights into the severe limitations of random mutation and natural selection. This slide lists 7 recent insights into the severe limitations of the mechanism of random mutation and natural selection. But before discussing each of these new insights, I'll review some basic background to explain the mechanism of random mutation and natural selection. I will review some background information for sexually reproducing organisms. The concepts are similar but simpler for non-sexually reproducing organisms. In sexually reproducing organisms, an individual receives a set of chromosomes from their mother and father. So the individual has 2 copies of each gene. In the process of producing the germ cells, egg or sperm, which contain only 1 set of chromosomes, the single set of chromosomes will contain a random combination of genes from the 2 parents, as indicated by the red and blue colors in this image. This produces genetic variation, but does not create new genes. In the standard neo-Darwinian theory, random mutations are believed to be the source of new genes or new genetic information. Richard Dawkins expressed this in his book, The Blind Watchmaker, as follows. He said, "Natural selection may only subtract, but mutation can add." Likewise, Douglas Futuyma wrote, "Mutation is ultimately the source of new genetic variations, and without genetic variations there cannot be genetic change. Mutation is therefore necessary for evolution." There are 2 classes of mutations. Point mutations occur when there's a change, addition, or deletion of a single nucleotide in DNA. These initially increase the number of versions of the gene, or in other words, the number of alleles. However, when many point mutations accumulate, then in principle, entirely new genes can be created. Chromosome mutations occur when a segment of a chromosome is duplicated, or deleted, or inverted, or transferred to a different position on the same chromosome, or to an entirely different chromosome. Chromosome mutations change the sequence of genes on affected chromosomes. The most commonly cited mechanism for the creation of new genes is gene duplication, followed by the accumulation of point mutations in one of the copies. The first recent insight into the severe limitations of the mechanism of random mutation and natural selection is that experimental evolution studies have shown very limited change. The most extensive laboratory evolution study was performed by Professor Richard Lenski and his research group at Michigan State University. They conducted a long-term evolution experiment with E. coli bacteria from 1988 until 2022. They started with 12 identical populations of E. coli. Each day, his research group would transfer a sample of each culture to a fresh culture of the same medium. The medium had a low concentration of glucose, 25 milligrams per liter, and included citrate at 11 times the concentration of glucose. The group periodically froze samples of the populations for genetic analysis. It's interesting to note that the research group started this project before rapid genome sequencing technologies were available, but they anticipated that technology would soon be developed. In 2022, the experiment reached 75,000 generations, which corresponds to about 1.5 million years for humans. But the experiment with bacteria involved far more organisms than could have occurred within early primate populations. At 50,000 generations, they sequenced 264 genomes. At that stage in the experiment, the following changes had occurred. They observed faster growth rate, but the improvements diminished over time. The faster growth rates occurred because some genes that were not needed were turned off, such as genes for making ribose or for constructing the flagellum. They observed hypermutators that had 100 times more mutations than the original organisms. This occurred due to broken DNA repair genes. 97% of the observed mutations occurred in these organisms. They observed larger cell sizes than in the original culture. At 50,000 generations, the average genome size had declined by 63,000 base pairs, or 1.4%. The most significant change that occurred was that some cells developed the ability to grow on citrate, a small organic molecule that was present in the media in excess. This graph from a research paper shows the rapid onset of the ability to use citrate that occurred at just over 33,000 generations. Normally, E. coli has the ability to use citrate for growth in absence of oxygen, but this ability is turned off when oxygen is present. In this case, a segment of DNA was duplicated, which caused the citrate transporter gene to be turned on in the presence of oxygen. This allowed a short-term increase in growth through the use of citrate in the presence of oxygen. But this disruption of the normal regulation of the cell eventually led to a greatly increased death rate, greater than 50%, described in detail in the articles shown here. In 2022, the experiment reached 75,000 generations. For humans, that many generations corresponds roughly to 1.5 million years. An analysis of human and chimpanzee genomes, reported in 2006, concluded that 690 new genes, including 20 entirely new protein families, arose within humans since the assumed divergence from a common ancestor 6 million years ago. Assuming that happened at a linear rate with time, then 1.5 million years would correspond to roughly 170 new genes, including something like 5 entirely new protein families. It seems that 75,000 generations is long enough to expect some new genes and proteins in the long-term evolution experiment. However, in this experiment with E. coli, the most extensive laboratory evolution experiment to date, no new genes or proteins occurred within the 75,000 generations. For some further information and discussion of this experiment, see the links shown on this slide. The second recent insight into the severe limitations of the mechanism of random mutation and natural selection is that in rapidly evolving groups, variation is limited to the level of species and genera. The chart shown here from Michael Behe's book, Darwin Devolves, shows that for various types of organisms that are known to have diversified rapidly, such as finches or cichlid fish or fruit flies, the diversification does not lead to new families but stops at the level of genera. He writes, "Minor random variations around a design blueprint are possible and can be helpful, but are severely limited in scope. For new basic designs, such as those at the biological level of family and above, additional information is necessary, information that is beyond the ability of mindless processes to provide." One example is the diversification of cichlid fish in Lake Victoria in Africa. This lake completely dried out at several times in the past, and the current lake is believed to have arisen about 15,000 years ago. Since that time, a dramatic diversification of cichlid fish has occurred. This is believed by some to be the most dramatic example of evolution in modern times. However, while about 500 species and 75 genera have developed, the diversification stops at the level of genera, and no new families have arisen. The third recent insight into the severe limitations of the mechanism of random mutation and natural selection is that genome sequencing has shown that diversification or microevolution often occurs by shuffling existing genes and by breaking genes. This new insight was made possible by the huge recent advances in sequencing technology. This chart shows the decrease in the cost of sequencing a human genome as sequencing technology has advanced over time. This new technology has enabled studies of the genomic changes that occurred during diversification events. The fact that mutation and selection most often operates by breaking genes is simply a consequence of the fact that, as for any complex object, far more individual changes will damage or disable genes, whereas only very few possible changes will improve them. Michael Behe summarized much of the evidence from large-scale sequencing studies in his book, Darwin Devolves. The results led him to propose what he refers to as the first rule of adaptive evolution, which is that the process of random mutation and natural selection will break or blunt any functional gene whose loss would increase the number of a species' offspring. You can find a more detailed discussion of this in this article by him in the Quarterly Review of Biology in 2010. As an example, he cites the following results in relation to various breeds of dogs. Increased muscle mass is due to degradation of a myostatin gene. Yellow coat color is due to the loss of function of the melanocortin-1 receptor. Black coat color is due to the deletion of a glycine residue from beta-defensin. Damage to 3 genes affects fur characteristics, either long or curly. Variation in size is due to 6 genes. Half involve degrading changes in the protein coding region and half in the control regions. All mutants decrease size. A short muzzle size is associated with decreased activity of the THBS2 and SMOC2 genes and a damaging point mutation to BMP3. Short tails are due to loss of function mutation of another gene. Richard Dawkins stated that dog breeds are a premier example of the power of selection by humans, not by nature, to shape animals. But Behe responds by writing that Dawkins is exactly right. Dog breeding is a wonderful example of the power of selection acting on hidden random mutations. But now that we can investigate the molecular level of life, we see that the great majority of dog mutations unwittingly selected by us, humans, are very likely to be damaging, degrading, or outright loss of function. Behe also highlighted the case of polar bear divergence from brown bears. Sequencing has shown that of the most strongly selected genes, half of the mutations were predicted to damage function. Specifically, mutations to the APOB gene, involved in fat metabolism in mammals, and to the LYST gene, involved in pigmentation, were predicted to be damaging. The conclusion is that polar bears adjusted to the harsh climate through the degradation of genes. Large-scale sequencing has also been performed for finches. Finches in the Galapagos are believed to have diversified into 14 species over a period of 1.5 million years. More recently, variation in the structure of the beaks during and after a drought has been cited as a remarkable example of evolution in action. The genomes of 120 finches from all species of the Galapagos were sequenced. The authors of this article conclude that hybridization, in addition to natural selection, played a major role in the diversification. The strong role of hybridization contrasts with the common observation that hybrids are less fit. Hybridization results in an increase in genetic diversity by combining genes or alleles from different species. In addition to identifying the surprising importance of hybridization, the study also identified a gene labeled ALX1, which is strongly associated with beak shape. 2 mutations seem to account for the changes in beak shapes. Computer analysis of those mutations classified them as damaging. From this, Michael Behe speculates, if the normal activity of the ALX1 protein during development helps make a beak sharper and more elongated, then hindering its activity could cause the beak to develop as less sharp and less elongated. In other words, shorter and blunter. So the adaptive nature of finch beak variation seems to arise from a damaged gene. The study reports no evidence of entirely new genes in the diversification. Finally, large-scale sequencing has also been performed for cichlid fish in the African lakes near Lake Victoria. This diversification was far more dramatic than that of the finches, in that in Lake Victoria, 500 species developed within 14,000 years, compared with 14 species of finches in 1.5 million years. This is considered the most spectacular example of evolution in modern times. One might wonder whether this very rapid burst of diversification occurred because of fortuitous mutations that led to new genes that enabled this adaptive radiation. This figure comes from a paper in 2014 reporting the genomic sequencing of cichlid fish from several lakes in Africa. They sequenced 5 species from different older lineages and 6 closely related species from a recent lineage. A more extensive study reported in 2023 sequenced the genomes of 464 cichlids from all major lakes in the region. The study included several species from almost all ecological groups in Lake Victoria. The results showed that the genes underlying adaptation in African Great Lake cichlids were present in their ancestral taxa. No evidence was reported for the role of new genes. One gene involved in the production of the fish body plan was discussed in the supporting information for the 2014 article, which had mutations that appeared to disable 2 functions. The fourth recent insight into the severe limitations of the mechanism of random mutation and natural selection is that mutations that act early in the development of animals are lethal. Mutations that act late in development may be tolerated in some cases, but they do not lead to fundamental change. This chart shows early stages of development, from fertilization through the initial cell divisions to blastula and then gastrulation and organogenesis. To change body plans, mutations must occur at the point in development where body plans are put into place, but this often occurs right at the beginning. Changes in body plan cannot be accomplished through the accumulation of late-acting mutations, but mutations that act early in development are typically lethal. In the following slides, I'll emphasize this point by noting some statements from books and articles on this topic. Wallace Arthur, in The Origin of Animal Body Plans, wrote this: "This approach has important consequences for the neo-Darwinian view that major morphological differences between organisms from different higher taxa have been produced through the gradual accumulation of very small changes. Specifically, while in a non-developmental approach it seems plausible that many small changes can indeed accumulate to give a larger one, in a developmentally explicit approach it is clear that many late changes cannot accumulate to give an early one. Thus, if taxonomically distant organisms differ right back to their early embryogenesis, as is often the case, the mutations involved in their evolutionary divergence did not involve the same genes as those involved in the typical speciation event, where usually the early embryogeneses of the daughter species concerned are virtually identical." Similarly, Carroll, in Patterns and Processes of Vertebrate Evolution, wrote, "There are significant differences between the nature of the majority of genetic differences studied by evolutionary biologists and of those that contribute to understanding the embryological patterns and processes of interest to developmental biologists. It is only late in development, when their anatomical patterns are nearly fully formed and the young are close to hatching or birth, that the traits commonly studied in classical genetics become evident. Miklos and Johns argue that macroevolutionary change requires changes in very early embryogenesis. This image from The Origin of Animal Body Plans illustrates this point. It shows developmental time on the x-axis and the probability of a mutation being advantageous on the y-axis. Carroll writes, "In general, the earlier a gene is expressed, the greater influence it can have on subsequent events during development, and the more likely that its disruption will result in malformation and death. Most mutations known to cause changes in developmental processes are too damaging to provide a model for the manner in which evolutionary advances may have occurred." John MacDonald expressed the issue this way: "Those genes that are obviously variable within natural populations do not seem to lie at the base of the many major adaptive changes, while those that seemingly do constitute the foundation of many, if not most, major adaptive changes apparently are not variable within natural populations." Eric Davidson was a pioneering developmental biologist at Caltech. He and his collaborator, Douglas Irwin, described the conundrum this way: "There's always an observable consequence if a developmental genetic regulatory network subcircuit is interrupted. Since these consequences are always catastrophically bad, flexibility is minimal. And since the subcircuits are all interconnected, the whole network partakes of the quality that there's only one way for things to work. And indeed, the embryos of each species develop in only one way." In 2011, Davidson wrote, "Neo-Darwinian evolution assumes that all processes work the same way, so that evolution of enzymes or flower colors can be used as current proxies for study of evolution of the body plan. It erroneously assumes that change in protein coding sequence is the basic cause of change in developmental program, and it erroneously assumes that evolutionary change in body plan morphology occurs by a continuous process. All of these assumptions are basically counterfactual. The neo-Darwinian synthesis from which these ideas stem was a pre-molecular biology concoction focused on population genetics and adaptation natural history, neither of which have any direct mechanistic import for the genomic regulatory systems that drive embryonic development of the body plan." The fifth recent insight into the severe limitations of the mechanism of random mutation and natural selection is that the waiting time for two coordinated mutations is now known to be prohibitively long for animals. Knowledge of mutation rates and population sizes for animals has led to the conclusion that genetic alteration will proceed one mutation at a time. This is a severe limitation because it seems inconceivable that irreducibly complex molecular machines or regulatory logic circuits could have been constructed one mutation at a time. In 2003, Orr pointed out that the severe limitation for the number of mutations for an adaptational step to occur was recognized and discussed by Maynard Smith in the 60s and 70s. Orr then states the issue as follows: given realistically low mutation rates, double mutants will be so rare that adaptation is essentially constrained to surveying and substituting one mutational step neighbors. Thus, If a double mutant sequence is favorable, but all single amino acid mutants are deleterious, adaptation will generally not proceed. Following this earlier work, Michael Behe evaluated the waiting time for two coordinated mutations using experimental data. He used the experimentally determined waiting time for two coordinated mutations for the malarial parasite to escape the drug chloroquine. He transposed this data onto the case of human populations, and the result was a waiting time of 10 to the 15th power years. The waiting time for two coordinated mutations was further examined with theoretical modeling. Such models require assumptions for mutation rates, population sizes, and for whether each mutation is beneficial or neutral. A specific example was analyzed by Durrett and Schmidt in 2008. They stated, "We now show that two coordinated changes that turn off one regulatory sequence and turn on another one without either mutant becoming fixed are unlikely to occur in the human population." In their paper, they went on to state that the estimated waiting time for two coordinated mutations in humans is about 162 million years. They wrote, "As our new results show, a coordinated pair of mutations that first inactivates a binding site and then creates a new one is very unlikely to occur on a reasonable timescale." The value they arrived at from their theoretical modeling was much lower than Behe's estimate based on real data. Behe responded by pointing out several flaws in their analysis that, when fixed, bring the modeling estimate more in line with the estimate based on real data. The waiting time for two coordinated neutral mutations in a hominid population was also analyzed by numerical simulation, which is an entirely different approach. The simulation was performed for a population of 10,000 individuals with a generation time of 20 years. They included some very conservative assumptions, such as that all evolving strings were neutral until the specified string occurred, and that once the target string occurred, it was protected from further mutations. Their results showed that as the string length increased linearly, the waiting time increased exponentially. To establish a string of two nucleotides required on average 84 million years. The authors include a discussion of aspects that might reduce the waiting time further. However, the authors concluded that even using the most generous feasible parameter settings, the waiting time required to establish any specific nucleotide string within this type of population was consistently prohibitive. For further discussion of this topic and various responses to the waiting time problem, see the links shown on this slide. The sixth recent insight into the severe limitation of the mechanism of random mutation and natural selection is that much of the information of any living organism, and one estimate is about 1/2 for animals, is not encoded in the genome. This insight is part of a revolution taking place in biology from a gene-centric view to a systems biology approach, or an organism-centric view. This article by British physiologist and biologist Denis Noble that appeared in the journal Nature in February 2024 illustrates this revolution in thinking. It emphatically states that genes are not the blueprint for life, as some biologists had proposed previously. In an earlier book titled Dance to the Tune of Life, Noble stated, "Something is happening in biology that is analogous to what quantum mechanics and relativity did in physics in the beginning of the 20th century. This is the revolution regarding the role of genes that I just referred to." He goes on to state that a cell is vastly more than its DNA, and that the information contained in the 3D cell structure is comparable to that contained in the genome. This insight is important for many reasons, but it's important for our current topic because if the information to build an organism is not in the genes, then genetic mutations cannot account for the developmental program. You might wonder, if genes are not the blueprint for life, what are they? What role do they play? The answer is that genes supply cells with materials. I'm not sure when this insight first emerged, but it's clearly stated in this article from 1990. The author states, "Genes contain no information about levels of organization higher than the primary structure of proteins. The simplest and also the only strictly correct view of the function of genes is that they supply cells and ultimately organisms with chemical materials." Dennis Noble stated elsewhere that the genome should be considered as a database. This is stated in these two quotes from an article in 2012. The coding sequences form a list of proteins and RNAs that might be made in a given organism. These parts of the genome form a database of templates. To be sure, as a database, the genome is also extensively formatted with many regulatory elements, operons, embedded within it. The genome is not a program, but rather a read-write memory that can be organized in response to cellular and environmental signals. The protein coding sequences are templates. They determine which set of proteins the organism has to play with, just as a child knows which pieces of Lego or Meccano she has available for construction. Those parts of the genome are best regarded as a database. Noble rejects such terms as selfish genes, genetic program, blueprint, book of life, and replicator. By the way, there's no such thing as a gene that replicates on its own. Gene replication requires the machinery of the cell with many protein components. These terms and the gene-centered view were strongly promoted by Richard Dawkins' book, The Selfish Gene, published in 1976. Which was one of the most influential science books of the 20th century. This view is now known to be incorrect. Compare building a body to building a house. Think about what it takes to build a house. It takes building materials, assembly instructions, and a floor plan. This new insight is that the floor plan is not in the DNA, and so DNA mutations cannot alter that. The final insight into the severe limitations of the mechanism of random mutation and natural selection is that evidence is growing for natural genetic engineering mechanisms of adaptation operating within a constrained design. This new insight indicates that organisms are even more complex than previously thought, and that organisms may have some ability to alter their own genomes. This new insight means that much of the adaptation previously attributed to random mutation and natural selection may be due instead to natural genetic engineering mechanisms of adaptation. James Shapiro has been a leading researcher on this topic. This slide shows his recent book, Evolution: A View from the 21st Century, that I recommend. I also highly recommend the short summary article that can be found at the link shown at the lower left. The severe limitations of random mutation and natural selection, or neo-Darwinism, has become an important topic of discussion in evolutionary biology. With this motivation, some highly accomplished scientists and philosophers have formed a group called the Third Way of Evolution. On their website, they state that there is no empirical basis for assuming that random mutation and natural selection is a unique creative force that solves all the important problems of biology. While recognizing the severe limitations of random mutation and natural selection, they strongly affirm that they do not support the notion of design from an intelligence outside of nature. This is why the group is called the Third Way of Evolution. One of the members of this group, Gerd Müller, a theoretical biologist from the University of Vienna, co-authored a book in 2003 called Origination of Organismal Form. In that book, he and his co-authors stated the following: the neo-Darwinian paradigm still represents the central explanatory framework of evolution, as exemplified by recent textbooks. Although this theory can account for the phenomena it concentrates on, namely variation of traits in populations, it leaves aside a number of other aspects of evolution. Most important, it completely avoids the origination of phenotypic traits and of organismal form. In other words, neo-Darwinism has no theory of the generative. As a consequence, current evolutionary theory can predict what will be maintained, but not what will appear. The authors further affirm that organismal form cannot be explained from genetics. They write, "Now that the entire genomes are mapped out and the genomic approach is seen to be unable to explain biological complexity, this problem will be a central concern of future research. Recognizing that the origination of biological form cannot be understood solely from genetic analysis will necessarily stimulate investigation of the processes that actually construct the phenotype from materials provided in part by the genotype." In 2016, a conference was held on this topic by the Royal Society of London called New Trends in Evolutionary Biology. In 2022, a conference in Israel was held on the potential and limitations of evolutionary processes. Another example that the severe limitations of random mutation and natural selection has become an important topic in evolutionary biology is this recent article in The Guardian, posing the question, do we need a new theory of evolution? To summarize, this new discovery is that random mutation and natural selection has severe limitations. This blind watchmaker mechanism that was once proposed to be able to explain the appearance of design in biology and to climb Mount Improbable, to reference two books by Richard Dawkins, does not have the tremendous creative power ascribed to it in the past. Rather, it's now known to be very weak, with little or no truly creative power.